Welcome friends, welcome to this video where we are going to look at biology paper 2 for the year 2020, and we are focusing on the paper that was written by the external candidate. So let's look at the first question from section A, where we have figure 1.0 shows a plant cell. Okay, then the first question that we are expected to answer here is, we have named the parts about A, B, and C. So A, we can see that this is the vacuum. And the function of the vacuum to store salt solution. Then the other question is B. So B is pointing at this shaded part here. So this is the nucleus. Then the nucleus has functions like to control all the reactions that occurs in a cell, to store genetic makeup for the cell. Then for C is cell membrane. So cell membrane is our C. Now Roman number 2, name the organ in the plant in which figure 1.0 is found. Give a reason for your answer. So the organ where this cell is found is a leaf. Then reason, this is where photosynthesis occurs. So if we look at photosynthesis, definitely we are looking at the process that is involved during the formation of glucose. Then we move on to question B. Describe two adaptations of the cell in figure 1.0 which enable it to carry out its function. So let's go back to our figure. So this cell, if you can see by its diagrammatical way, we can tell that we are dealing with a palisade, and the palisade, the main function of the palisade is to carry out the process of photosynthesis. Now, if you look at the adaptation, we are looking at the characteristics or some of the features that were put in place to make sure that it does this process. So the first one, from the structure, we saw that it has numerous chloroplasts. So because of this, the rate of photosynthesis is high. Then the other adaptation that we can look at here, we can say the cells are longer vertically than horizontally. So this statement means that here we can say that in this vertically they are longer, then here they are shorter. Now, what is the importance of this? It enables this allows chloroplasts to migrate upward or downward as light intensity changes, so that they are not damaged by the light. So the chloroplasts are able to move upward or downward. It depends on the amount of light. So if there is too much light, they will move down to be protected away from the too much light. So that is the importance of being longer vertically. So these are the two most important adaptations that you need to look at whenever you are dealing with a palisade. Remember, the main function of the palisade is to carry out the process of photosynthesis. Now see, explain what would happen to the cell in figure 1.0 above if it was placed in a test tube containing distilled water. So it would gain water gain water by osmosis, and as the result, it will reach its highest target or turgidity. So a turgidity is a situation where the cell gained water, as a result it expands in size. So this happens only with the plant cell, because if you look at the animal cell here, the animal cell, if the animal cell gains a lot of water, it bursts eventually. Now for this one, we cannot expect this cell to burst because of the cell. So the cell helps to keep it in place. Now let's dive into question 2. Now under question 2, we can see that we have a tooth. So figure 2.0 shows the internal structure of a tooth. So it is very important, as you are studying, you also have to look at the structure. And to my journey of school journey, I've come to see that the biology is all about the diagram. So as you are studying, make sure you understand and you master the diagrams, because most of the questions comes based on the diagram, and you can make an explanation based on the diagram. So like the one here that we are given is a tooth. Now a tooth has so many parts that we have to look at here. So there is part F, D, E. Now let's look at the questions. Name the parts label D, and E in figure 2.0. So D, we can see that these are nerve cells. The nerve cells, we are looking at the cells that are responsible for the transfer of pain signals. Then for E, we are definitely looking at the dentine. So we have the dentine. Then the function of the dentine, it is just it contains the fine canals which link up the pulp cavity. Then the pulp cavity is just the space where we have these nerve cells, including the blood cells. So we are saying D is nerve cells. So we have nerve cell. Then E, we have dentine. Then B1, explain what happens to the part labeled F during tooth decay. So we would say there remains from the food that contains too much sugar will develop into an acid called plaque. So there is an acid that we call plaque. Then what this plaque does, it dissolves the enamel. So an acid called plaque or plaque. So this acid is there because of the remains from the food that we eat. Then after this changes into an acid, then it will start to dissolve the part labeled F, which is the enamel. So let's say this acid will dissolve part labeled F, resulting in a tooth decay. So the other thing, then you are saying tooth decay, sometimes it's also called dental decay or dental caries. So that is the other name. So this is just the condition where the enamel of the teeth is dissolved or corroded by organic acid produced by the fermentation of sugars by bacteria in the mouth, forming cavities in the teeth. So that is what we are saying. So this tooth decay, we are also saying it's also a dental decay, because sometimes it may be given such language. So dental decay or dental caries, so it's the same thing. So if the question explains this, just know that you are dealing with the tooth decay. Then looking at the shape of the tooth in figure 2.0 above suggests the role it plays in the digestion. So in the digestion, we are definitely looking at physical digestion, because physical digestion has to do with the breaking down of food by physical means, so with the help of the teeth. So by looking at the shape, we can tell that this should be a molar or premolar. So the function, we can say it plays a role in physical digestion. To grind, chew, and the crushing of food. Okay, so now we have this question here. Complete the human dental formula below by filling in the missing numbers G, H, and J. So let's look at the first one. These are scissors, then H. So H should just be 1. So remember, if you are given the dental formula for human being, the number of the canine on top is equal to the number of the canine on the lower jaw. So since we have 1 on the upper jaw, then 1 also on the lower jaw. So our H is equal to 1. Then what's our J? Remember, the picture here is not for O, but for a human being. So the total is adding up to 32. Now let's look at the molar. So if we have 3 on the lower jaw, how many of these can be on the upper jaw? So we are still going to have 3. Then our, okay, I think let's not see. So J is 3, H is 1. Now G, we are looking at the number that we can multiply by the total of these to give us 32. So remember, if there is 2 on top, so 2 plus 2, here we have 4. 1 plus there is 1, so here there is 2. 2 plus 2, here there is 4. 3 plus 3, here there is 6. So 4 plus 2 is 6, plus 4, 10, plus 6, that is 16. So this is same as G times 16 is equal to 32. So to solve for G, we divide by 32. So G is equal to 2. So this means that G represents the number that we can multiply by the numbers inside to add up to 32. So it is just the 2. And I'm sure we all remember this pattern of you count the total number, then you multiply by 2. Now we move on to another question. Figure 3.0 shows a section through the nephron. So if you look at the nephron, we are looking at the microscopic or the functional unit of a kidney. So this is where the urine formation And the processes under urine formation, we know we'll have pressure filtration, selective reabsorption, and also osmoregulation. So all these processes plus some of what I've not mentioned occurs in a nephron. Now, let's look at some structure. So there is this J, K, then we have L. So now let's look at the first question. Name the parts labeled J, K, and L. So let's look at our J. So J is this part here. So this is glomerulus. Then what of K? So K is this part which is the Bowman's capsule. Then L, the vessel that carries the urine from the nephron to the bladder. I mean, before to the bladder is the ureter, but before ureter we have what we call collecting duct. So these collects from thousands or billions of nephrons. Then they join, formally forming what we call the ureter. So collecting duct is our L. So suggest 3 substances that are not filtered out at J. So what is J? J, we say it is glomerulus. So the first one, proteins. Why? The sizes. So they are not able to pass through the pores that are found on the glomerulus. Then number 2, red blood cells. Number 3, white blood cells. So the picture at this point is you just have to look at the materials that are in larger sizes. Then explain what would happen at J if the coiling of the blood capillary was increased. So the coiling is like this. If we increase the coil, then the pressure filtration, so much of the waste product will be filtered out into the Bowman's capsule. So we'll say this would increase the process of pressure filtration. Which is it? So most of the waste product will be removed. Then name the hormone that causes the reabsorption of water in the kidney and the organ where it is produced. So the hormone is antidiuretic hormone, which is also known as vasopressin. Then this is initialized by ADH. The name of the organ is just the brain. Don't say the pituitary gland, that's just a gland. Now the question is the organ, so it is the brain. Then we move on to question 4, figure 4.0. Below shows the external parts of the human eye. Okay. Then name the parts labeled M and N. So what's our M? M is the pupil. Then N, this is the cornea. Then explain how N is adapted to its function. So if you look at the cornea, we are looking at the refraction of light. So we'll say its adaptation is all about the transparency and the way it is made. So it is made from a tough fibrous, because of that it is able to reflect light, or it helps light to bend so that we may have a clear picture. So we say it is made up of tough, so say tough transparent material which helps light to bend so that we have a clear image. So for us, or maybe animals to have, or maybe to have an image, light must bend, it has to reflect. That's why we have the lens. So if you look at science, lens is any object that helps light to bend, and because of that an image is formed. B, describe the role played by M and O when a person moves to a room with a dim light. So to a dim light, definitely we are looking at where there is a low light. So M is a pupil. So what happens to the pupil? So we know that it will dilate. So this will dilate to allow more light to enter. Then O, let's go back. What's structure O? So structure O is should be iris, because yeah, that's iris. So the iris contains two muscles that are important. Then in these muscles we have the circular muscle and the radial muscle. These are the muscles that are responsible for the changing of the size of the pupil. So remember, the pupil changes its size depending on the amount of light that the eye is receiving. So say this contains muscles which will contract and relax to make part M dilates. Okay, then you see, explain the cause of short-sightedness and how it can be corrected. So if we have this is our eyeball. Let me see if I can give you a picture here. So let's say we have an eyeball. So the most important part in terms of formation of the image is the part known as the retina, and it's somewhere here. So if the rays of light are coming in this direction, so there are some times where the normal vision, they have to meet at the retina. So this is where they are supposed to meet so that we have the clear picture. Now what happens if these rays of light meets before retina? Then we have the short-sightedness, because long-sightedness, the rays of light meets beyond the retina. So before retina we have short-sightedness. So we say causes, this is when light rays meets before retina. So meets before retina. So this can be because there's too much, the vitreous humor has too much strength. As a result, it is making light to bend more, or maybe there is an enlargement of the eyeball. As a result, light cannot meet on the retina. Okay, so then correction. So correction will say by wearing or using glasses containing glasses containing concave lens. Let's move on to our last question for this video. So red-green color blindness is a sex-linked characteristics. A1, define sex-linked characteristics. So these are characteristics that are carried on the X chromosomes. Then what can cause sex-linked genetic disorders like the red-green color blindness? So here, this is just the condition where a person with such a problem may fail to differentiate the difference between red and green. For example, you give this person these two colors and you tell him or her to differentiate. That person may not see the difference, which one is red and which one is green. So that is the problem with these people. So that is what the problem is. So now the causatives for red-green color blindness, they can be caused by the genetics. We know that it's genetics, but what are those? Then in terms of so this can be perceived green deuteronomy and a difficult perceiving red protonomy. Now in a simpler way, we can just say that it's a condition where the receptors that are responsible for color have defect. So we know how do we see that this is red, how do we see that this is blue, green. So it's all about the light that comes with all the colors. We know about Roy GBV. So now the substance has the ability to absorb some of the light that it is receiving. Now the point is the person with such may find it difficult to differentiate because of the receptors that are responsible. The receptors that are responsible for this cannot tell or cannot receive these two colors. Now, how can we summarize? So we say, we can just say what causes sex-linked. So we'll just say gene cataracts. So we can call this as or sometimes this point cataract is like a cloud that forms. Now this cloud is just only covering the cells that are responsible for colors. As a result, someone cannot see the colors. A normal couple had a colorblind son using the gene error from normal color vision and a gene error for color blindness. Write down the genotypes of the first one further. So the father must have the first one, no? XY. Then if you just hear a normal couple had a son who is, it may be the same that we are looking at color blindness, or maybe hemophilia, sickle cell anemia. So any of these sex-linked characteristics, the point is, if the question is a normal couple, just know that the man is okay. And whenever the man is okay, you put capital letter R to the X. Y does not take these characteristics. Then the mother will have XX. Remember, for this couple to have a son who has this condition and they are considered to be a norm, just know that the mother must be a carrier. So a carrier, we are going to have one capital letter and one small letter. So the point is, a man is either okay or sick. There is nothing like a carrier when it comes to men. So if the couple is normal and they happen to have a son with this condition, then just know that the mother is a carrier. So that's the point that you. Then using a genetic diagram, show how the son could have been born color blindness. Okay. So say parental generation P1. Then what do you do? So we can now separate. Then if we separate, we can say gametes of P1. Then from there, we are going to have now random fertilization. So say random fertilization. So random, it means we don't know which chromosome will meet with the other one to, the gamete. So we can now see that here we have a baby girl who is 100% okay. Okay, so there is a chance for this couple to have a carrier. There is a chance to have a son who is 100% okay. There is a chance to have a son who has ready green color blindness. So this is how that problem came into existence. So say genotype. So genotype of F1. So this is the picture that we can look at under this paper. So that's it for this video. Thanks for watching.